So let's come on the next topic, that is mechanism of cell injury. So here is a list of the university exam questions which are usually asked from this topic. So I request you to look at the university exam questions either in the notes or it is provided in the PYQ sections also. So you just open it there and have a look, and whatever I am discussing, all the questions, the long, short and very short will be covered there. So coming on the mechanisms of cell injury. Basically, there are 4 mechanisms of cell injury. I told you whenever all the cells in human body are in homeostasis. So whenever the stress is acting, 3 things happen one by one. The first thing is the cell adaptation. We have already covered that. Now the reversible and irreversible cell injury. So what are the mechanisms of reversible and irreversible cell injury? There are 4 mechanisms of these injury. The mechanisms of reversible and irreversible cell injury. So the first mechanism is ATP depletion. The second is mitochondrial damage. The third is loss of calcium homeostasis, and finally free radical injury. Most of the PYQs which are asked in university exam are on free radical. Free radical is very important. You get many questions on that. So I will be discussing all 4 one by one. The first one is the ATP depletion. So have a look in the diagram. Can you see this is a cell? This is a normal cell of human body. You can see inside the cell there is a nucleus. This is the mitochondria. This is endoplasmic reticulum on which the ribosomes are present. You can see the ribosomes are present. And you can see this is sodium potassium pump which is present on the cell membrane. This is a normal cell. It is getting the blood. Now imagine if this cell do not get the blood. That is, ischemia is happening to this cell. What will happen? Why the blood is required by all the cells? From head to toe, all the cells require blood. Why the blood is required? From the blood, the cell get oxygen. The oxygen goes inside the mitochondria, and in the mitochondria, the oxygen get converted into ATP. I mean, the ATP is synthesized in the mitochondria, and the mechanism is known as oxidative phosphorylation. In presence of oxygen, the ATP is synthesized in the mitochondria. That's why mitochondria is known as powerhouse of the cell. Now, if the cell is not getting the blood, the cell is not getting the oxygen, the cell is not getting the oxygen inside the mitochondria, there is no oxidative phosphorylation. If there is no oxidative phosphorylation, there is no ATP synthesis. So that is the mechanism ATP depletion. You will be thinking, ma'am, why ATP is required for the cell? For the cell, if ATP is not there, will the cell injured? Yes, the cell will have cell injury if ATP is not there by 3 mechanisms. So let me explain you the 3 mechanisms. What will happen because of ATP depletion? So first you have to draw this flowchart, and after that you have to combine 3 flowcharts here because of ATP depletion. What is happening? So the first thing which is happening due to ATP depletion. Now here there is ischemia. I told you the cell is not getting the oxygen, the cell is not getting the blood, so ATP is not synthesized in the mitochondria. This ATP, normally the ATP is required for the functionality of sodium potassium pump. For the functionality of sodium potassium pump, ATP is required. Now if there is no ATP, this pump is not functioning. Got it? This sodium potassium pump is not functioning. Normally sodium goes out and potassium comes in when ATP is present. This is happening normally. You can see the zoom version of this pump here. You can see this is the cell membrane. You can see sodium is going out. Look at the arrow. And you can see the potassium is coming in. Look at the arrow. This is normal. But imagine if ATP is absent, this will not happen. This is not happen because the pump is not functioning now in absence of ATP. Because there is ATP depletion, there is ischemia. Ischemia means no blood. No blood means no oxygen. No oxygen means no oxidative phosphorylation. No oxidative phosphorylation, no ATP. ATP is not there. The pump is not functioning. So sodium will not go out. Sodium will remain inside the cell. And potassium will not come in. Potassium will present outside the cell. So there is influx of sodium and efflux of potassium. The sodium is present inside the cell only, and potassium is present outside the cell. They cannot go inside and outside, like the pump is not functioning. And you know, water follows sodium. There is a rule. Wherever the sodium will go, the water will go there. So I'm saying the sodium is not going out. So water is also not going out. Normally sodium and water goes out, but here they will remain inside the cell and they will swell the cell. Now because sodium and water is not going, they will swell the cell. The cell will swell. Inside the cell, endoplasmic reticulum will also swell. Endoplasmic reticulum will also swell. The cell will swell. The endoplasmic reticulum will swell, and on the surface of the cell, small blabs will come. You know, these are the blabs I am drawing. On the surface of the cell, the blabs will come. So this is how the cell get injured. This is the first mechanism due to ATP depletion. The sodium potassium pump is not functioning. The second thing, again, there is ischemia, no blood, no oxygen, no ATP synthesis in the mitochondria. Now, cell will try to synthesize ATP without oxygen. That is anaerobic glycolysis. Anaerobic glycolysis. You know, in aerobic glycolysis, ATP is synthesized in the mitochondria in presence of oxygen. So it is a better way. More ATPs are synthesized. But if oxygen is absent, cell will try to compensate and try to make ATP in absence of oxygen. That is anaerobic glycolysis. You may have read this in biochemistry. Here less ATPs are synthesized, but you may have seen the end product in biochemistry here is the lactic acid. So here the end product is lactic acid. Lactic acid will cause the pH of the cell acidic. The pH of the cell will fall. It will become acidic. And in acidic pH, the nucleus of the cell will form the clumps. Inside the nucleus, small clumps are formed. So nucleus is gone. Till now, what has happened? Because of the first mechanism, the cell is swelled. The endoplasmic reticulum is swelled. On the surface of the cell, there are blebs. And now nucleus is gone. The nucleus is gone. There is clumping of the nucleus because of this mechanism. And the third thing, again, there is ischemia, no blood, no oxygen, no ATP synthesis. Since there is no ATP synthesis, this is the endoplasmic reticulum. There are two types of endoplasmic reticulum, the rough and the smooth. You may be knowing that. The rough, the ribosomes are attached on the surface of endoplasmic reticulum. And in smooth, the ribosomes are not attached. Ribosome is the main organelle in which the protein synthesis takes place. You may be knowing the protein synthesis takes place in the ribosome only if the ribosome is attached with the endoplasmic reticulum. If ribosome is not attached with endoplasmic reticulum, the protein synthesis will not take place. And for the attachment of the ribosome with the endoplasmic reticulum, ATP is required. In absence of ATP, they will detach. The ribosome detached from the endoplasmic reticulum and no protein synthesis in the ribosome. So this is endoplasmic reticulum. You can see on the surface of the endoplasmic reticulum, the ribosomes are attached if ATP is present. If ATP is not present, the rough endoplasmic reticulum will lose the ribosome. And ribosome will not form the protein. No protein synthesis inside the cell. Protein is required for the cell membrane, for cytoskeletal, for enzyme. No protein means no enzyme, no cell membrane, no cytoskeletal, everything is destroyed. So the cell get injured by three mechanisms. This is the summary. What is the summary? You can see. Start with ischemia. If there is ischemia, means no blood. No blood means no oxygen. No oxygen means no oxidative phosphorylation in the mitochondria. No oxidative phosphorylation, no ATP. That is ATP depletion. And because of ATP depletion, three things happen. What are the three things happening? You can combine the three flowcharts together in your exam copy. What three things are happening together? Number one, sodium-potassium pump will not function. Sodium-potassium pump will not function. So sodium will not go out, it will remain in. And water will also remain in. So sodium and water will remain in. They are not going out because sodium-potassium pump is not functioning. So they will swell the cell. The cell will get swelled. The endoplasmic reticulum will also get swelled. And the blebs will be there. On the surface of the cell, the blebs is there. This is the first thing. Now, in absence of ATP, this is the first thing. The second thing is anaerobic glycolysis. Anaerobic glycolysis. So here, due to anaerobic glycolysis, the lactic acid is formed. Lactic acid will reduce the pH. It will cause the acidic pH. And because of reduction in pH, acidic pH, the nucleus get clumped. The nucleus get plumped. The nucleus is gone. Now, the third thing that will happen in absence of ATP, the ribosome will detach from endoplasmic reticulum. They will detach. So they will not functional. They will not synthesize the protein. No protein synthesis. No protein synthesis means there is no enzyme, no cell membrane, and no structural proteins. So imagine a cell. Can you imagine a cell in which the cell gets swelled? Because of swelling, the cell burst and died. And the endoplasmic reticulum is also swelled. Imagine a cell on which the blebs are there, in which the nucleus is gone, it is clumped. There is no enzyme, there is no cell membrane, it get destroyed. So this is the mechanisms of the cell injury. This is how the cell get injured. So this is the first mechanism. Everything is due to ATP depletion. Because of ATP depletion, I want you to combine the three flowcharts together. Because of ATP depletion, the three things are happening. You can see the same thing. Because of ATP depletion, you can see there is ATP depletion. The three things are happening. Sodium potassium pump is not functioning, the first thing. Anaerobic glycolysis, the second thing. And the ribosome get detached from the endoplasmic reticulum, the third thing. So combine the three flowcharts together. Draw flowcharts in your exam. Don't write paragraphs. If you want to get maximum marks in your university exam, please draw flowcharts and diagrams. Don't write paragraphs. The second mechanism is the mitochondrial damage. Now imagine this is the mitochondria of the cell. In the mitochondria, there are two membranes, inner mitochondrial membrane and outer mitochondrial membrane. And between the two membranes, there is a molecule which is known as cytochrome C, which is present between the two membranes. You know, mitochondria is the powerhouse of the cell. Why does powerhouse of the cell? It synthesizes ATP. I told you it synthesizes ATP. This is known as oxidative phosphorylation. So if the mitochondria is normal oxidative phosphorylation takes place, that is ATP is formed. Now the mitochondria get injured because of the injurious agent, the mitochondria is gone. It is non-functional. So three things will happen. Now, three things will happen again. Number one, normal oxidative phosphorylation will not take place. If normal oxidative phosphorylation will not take place, ATP will not form. And abnormal oxidative phosphorylation will take place. You know, normally, see. Here the oxygen is going. In presence of oxygen, ATP is formed. This is normal oxidative phosphorylation. In presence of oxygen, ATP is formed. This is normal. But this is not happening here because mitochondria is damaged. So ATP depletion is the first thing. And because of ATP depletion, again, the same three things are happening. You can interconnect. Okay. The second thing, abnormal oxidative phosphorylation takes place in which oxygen is not converting into ATP. Here the oxygen get converted into free radicals. So free radical formation takes place due to abnormal oxidative phosphorylation. And the third thing, because of mitochondrial damage, the cytochrome C will leak out. And that will cause the apoptosis. Let me explain. Okay. So I told you because of mitochondrial damage, this mitochondria is gone. It is damaged by the injurious agent. The injurious agent can be anything. The normal oxidative phosphorylation do not take place. Normal oxidative phosphorylation do not take place, so ATP will not form. Normally ATP is formed. Here ATP is not formed. So ATP depletion leading to the same three things again. You know, sodium potassium pump will not function, anaerobic glycolysis, and ribosomes will detach. The same three things you can write here. Okay. The second, normal is not taking place. Normal. So what is taking place? Say, ma'am, abnormal is taking place. So normal is not taking place. It is leading to deprivation of ATP. And abnormal is taking place. So here oxygen don't get converted into ATP. So oxygen get converted into what? Oxygen get converted into free radicals. That is reactive oxygen species of free radical. And here it will cause the deleterious effects on the cell. So inside the mitochondria, normally oxygen get converted to ATP. Here it is not getting converted. Here oxygen is getting converted into reactive oxygen species or free radicals. That is causing deleterious effects. So normal oxidative phosphorylation do not take place, abnormal take place. And the third thing is, you know, the cytochrome C, which is present between the two membranes of the mitochondria, it will leak out. Once the cytochrome C will out, it is coming out, it will cause apoptosis. The cell will do the apoptosis. It is the cell suicide, the cell will die. So these are the three things happening because of mitochondrial injury. What are the three things happening? Normal oxidative phosphorylation do not take place, and that is lead to ATP depletion. Abnormal oxidative phosphorylation takes place, and that will lead to formation of reactive oxygen species, that is free radical. And the leakage of the cytochrome C leads to apoptosis. So that is the summary of the second mechanism. So that is a summary. So same diagram is shown to you. Normal do not take place, abnormal take place, and leakage of the cytochrome C. That will lead to apoptosis. So that is the second mechanism I taught you, and I taught you the three consequences here. So I taught you the first mechanism and we have seen the three consequences here. We have seen the second mechanism and three. So I want you to draw the three flowcharts together here. I want you to draw the three flowcharts here. You will revise in the end. Okay. Coming on the third mechanism, that is loss of calcium homeostasis. What do you mean by loss of calcium homeostasis? See, this is a cell. Where is calcium? Calcium is very less. 99% calcium is extracellular. It is present outside the cell, not inside the cell. A very minute amount of calcium present inside the cell, a very minute 1%, but that too not in cytoplasm. So where does the calcium is present? Inside the cell, although very small, but it is sequestered in the mitochondria and endoplasmic reticulum. So calcium is present outside the cell, maximum, let's say 99%. Only a minute amount is present inside the cell, but that too not free. That too present either in the mitochondria or endoplasmic reticulum. Now, whenever the injurious agent is coming, see, injurious agent, so two things will happen. Number one, the extracellular calcium will go inside. The calcium channels will open and extracellular calcium will go inside the cell. And number two, intracellular calcium, which is sequestered in mitochondria and endoplasmic reticulum, that will also leak out. So in the cytoplasm, there is a storm of the calcium, abundant calcium in the cytoplasm coming from two places. One from extracellular and from the sequestered stores also. Okay, that is mitochondria and endoplasmic reticulum. So in the cytoplasm, abundant of calcium is there. You will say, ma'am, what is the problem if calcium is present in the cytoplasm? Is there any problem? Yes. Calcium acts as a cofactor for 4 enzymes. The 4 enzymes get activated in the cytoplasm. Which 4 enzymes get activated in the cytoplasm? Number 1, phospholipase. Number 2, protease. Number 3, endonuclease. Number 4, ATPase. Normally, these are inactive. But once the calcium is present in the cytoplasm, this calcium causes activation of the 4 enzymes. What does these enzymes will do? The phospholipase degrade the phospholipid. That is the cell membrane. Cell membrane of the cell is gone. Protease will degrade the protein. The proteins of the cell is gone. Endonuclease will degrade the DNA. So DNA, the nucleus is gone. And ATPase will degrade the ATP. So ATP is gone. So imagine a cell in which the cell membrane is gone, the protein is gone, the nucleus is gone, and the ATP is gone. So this is the mechanism of the cell injury. The cell get injured by multiple mechanisms. So this is the loss of calcium homeostasis. So normally calcium is intracellular. Normally calcium is extracellular. Extracellular is high. Intracellular is very low. Very low. If it is intracellular also, it is sequestered in mitochondria and endoplasmic reticulum. So whenever there is an injurious agent, two things will happen. The extracellular calcium will come inside, and the intracellular stores, that is sequestered calcium, will also come that from the mitochondria and the endoplasmic reticulum. So in the cytoplasm, there is abundant of calcium. That will cause activation of 4 enzymes. Which 4 enzymes? Can you tell me the name of the 4 enzymes quickly? What are the 4 enzymes? Phospholipase, protease, endonuclease, ATPase. What does they do? What does they do? Phospholipase degrade phospholipid, that is cell membrane of the cell is gone. Protease degrade the protein is gone. Endonuclease degrade the DNA, so nucleus is gone. And ATPase degrade the ATP, so ATP depletion, again the 3 deleterious effect. So in this way the injury happens. This is the third mechanism. So here also you have to write down the name of the 4 enzymes which get activated because of the calcium. You know the name of the 4 enzymes. So here I taught you the 3 mechanism. Here also I taught you the 3 mechanism. Here I taught you the name of the 4 enzymes you have to learn. And finally the ultra important, the 5-star important is the free radical injury. So coming on the last one, the free radical injury. It's ultra important. If free radical injury coming in your exam, the long short question, you have to write down under following headings. The first thing you have to write down the introduction. That is the definition of the free radical. What is a free radical? Then you have to tell me the generation. How does they get generated? Then tell me the types. How many types of free radicals are there? What does it causes in the cell, and how does the God has given the defense mechanism to remove the free radicals? So can you see the cell? Can you see the cell? Inside the cell, this is the mitochondria. Okay, now inside the mitochondria, what does happen? I told you inside the mitochondria, oxidative phosphorylation takes place. You'll remember what is oxidative phosphorylation, which is taking place on the inner membrane of the mitochondria. What does it mean? Here the oxygen get converted to water, and meanwhile ATPs are formed. Multiple ATPs are formed here. So how does oxygen get converted into water? What are the intermediate steps? So please see, these are the intermediate steps. So during the intermediate steps, the free radicals are formed. Can you see this is oxygen? Say yes. Can you see this getting converted into water? So when oxygen get converted to water, you can see 3 intermediates are formed. What are the 3 intermediates? The first intermediate is superoxide, that is O2 negative. This is superoxide, the first intermediate. The second is hydrogen peroxide, that is H2O2, hydrogen peroxide. And third is OH negative, that is hydroxyl ion. And finally, it get converted to water. These 3 intermediates are known as free radical. What is a free radical? Free radical is something that is having, it is a chemical, it is an intermediate chemical which is having single unpaired electron in their outermost orbit. Any molecule have single unpaired electron in their outermost orbit is very reactive. That's why free radical are also known as reactive oxygen species. It is one and the same thing, because it is having single unpaired electron in their outermost orbit. We are getting it. We are getting it. So they are formed in the mitochondria, the inner membrane of the mitochondria. They are very reactive because they have single unpaired electron in their outermost orbit, and they are formed as an intermediate during oxidative phosphorylation. So imagine a cell. Imagine the mitochondria of the cell. Imagine the inner membrane of the mitochondria where oxidative phosphorylation is taking place. In the oxidative phosphorylation, the oxygen get converted to water. But here the 3 intermediates are formed. 1, 2, and 3. The 3 intermediates are formed. You have to tell me the name of the 3 intermediates. What are the name of the 3 intermediates? First, superoxide is formed. That is O2 negative. Then hydrogen peroxide is formed, that is H2O2. And then hydroxyl ion is formed, that is OH negative. Learn their formulas, learn their names. So what are the 3 types of free radicals? The first is superoxide, second is hydrogen peroxide, and third is hydroxyl ion. These are the 3 intermediates of oxidative phosphorylation. In oxidative phosphorylation, the oxygen get converted to water, but it is not direct. The 3 intermediates are formed. It is formed in all of our cells. From head to toe, there are millions of cells. Inside all these cells, in the mitochondria of all these cells, this is happening. So in all human body cells, these are formed. These are formed as the intermediate species. These are intermediate chemicals. they all have single unpaired electron in their outermost orbit. This is their definition. That's why they are highly reactive. That's why they are also known as reactive oxygen species. So write down the definition that, ma'am, during oxidative phosphorylation in the mitochondria, when oxygen get converted to water, three intermediates are formed. That is, what are the three intermediates? Superoxide ion, hydrogen peroxide, and hydroxyl ion. These two are ions. They have charge on them. Okay. So total three free radicals are formed. What is the definition? They all have single unpaired electron in their outermost orbit. That's why they are very reactive. They damages the cell. Okay. So this is the definition. This is how they get generated. What are the types? I told you there are three types. What are the three types? Superoxide is O2 negative. Hydrogen peroxide is H2O2. And hydroxyl ion is OH negative. You can see this followed by this, followed by this. Okay. Now, how does this conversion take place? How does H2O2 get converted to OH negative? Can you see? H2O2 get converted to O2 negative in presence of iron. Where ferric get converted to ferrous in presence of iron. It is known as Fenton's reaction. It is known as Fenton's reaction. In Fenton's reaction, H2O2 get converted to OH negative in presence of iron. If it takes place in presence of oxygen, the same reaction, H2O2 get converted to OH negative only, but in presence of oxygen, it is known as Haber base. So I'm repeating. Please everyone concentrate. So ultimately, imagine a cell, any cell of human body from head to toe. Imagine any cell. Inside the cell, imagine the mitochondria. Imagine the inner membrane of the mitochondria where oxidative phosphorylation is taking place. Okay, where the oxygen get converted to water. Okay, oxygen get converted to water by combining with hydrogen. But there are three intermediates. So intermediates are the free radical. What are the three intermediates? You have to tell me the names. What are the three intermediates? The first intermediate is superoxide, O2 negative. Second is hydrogen peroxide, H2O2. And third is hydroxyl ion, that is OH negative. Now I am interested in this conversion, the second conversion. How does H2O2 get converted to OH negative? There are two mechanisms. It can take place in presence of iron, or it can take place in presence of oxygen. So there are two mechanisms. If it is in presence of iron, it is known as Fenton reaction. Fenton reaction. You get a very short note on that. So you have to write down this conversion in presence of iron, where ferric get converted to ferrous. If it is taking place in presence of oxygen, it is known as Haber base. Haber base reaction. Haber base. In both of them, the thing is same. What is the thing? That is H2O2 get converted to OH negative. But in presence of what? So same thing. You can see here also H2O2 get converted to OH negative. Here also H2O2 get converted to OH negative. But in presence of what? Is it presence of iron? Is it in presence of iron? If it is in presence of iron, it is known as Fenton's reaction. If it is in presence of oxygen, it is known as Haber base. So that is the thing you must understand. Now, whatever is the free radical, there are three radicals. They all inside the cell, they are generated. They damage the proteins in the cell, lipids in the cell, carbohydrate in the cell, nucleic acid in the cell. They damage the cell completely. So this is what they cause. They are very reactive. They damage everything in the cell. So you may be thinking that, ma'am, you are saying that from head to toe, we have millions of cells. Imagine the millions of cells inside your body. Inside each cell, in the mitochondria of each cell, these three free radicals are generated. And you are saying these three free radicals are highly reactive and they are damaging the cell. So our cells are not damaged. Why our cells? Now this cell, inside each of my cell, the three free radicals are generated because each cell of my body is conducting oxidative phosphorylation. So inside the mitochondria, these three free radicals are generated. But why my cells are not damaged? They are reactive. They should damage complete my cells. But our cells are not damaged because God is great. God has given three scavengers, which whenever the free radicals are generated, they are destroyed by the three scavengers. God has provided the three scavengers in all the cell. The three enzymes. What are the name of these three enzymes? Superoxide dismutase, SOD. That is known as SOD, superoxide dismutase. The second is glutathione peroxidase, GP. Glutathione peroxidase, and third is catalase. So these are the three enzymes present in every cell of human body. You can see this is the cell of any cell of the human body. This is the mitochondria. Inside the mitochondria, these three free radicals are generated. Here O2 negative is also present, H2O2 is also present, and OH negative is also present. But the cell contain the three enzymes, and these three enzymes will destroy the three free radicals. That's why our cells are not damaged, because thankfully we are having these scavengers. If we don't have the scavengers, you can imagine the free radicals are anyways they are going to generate inside the cell, because they are the intermediate species. So we are having the three enzymes, along with that certain vitamins like vitamin C, vitamin E, transferrin, ferritin, and celluloplasmin. They also act as a scavenger. But there are three main enzymes. What are the three main enzymes? Superoxide dismutase, the first one, glutathione peroxidase, the second one, and catalase, the third one. These are scavenger of free radical. They destroy free radical. So how does they destroy? Tell me the mechanism of all three. The first one, superoxide dismutase. You can see this is superoxide dismutase. It converts oxygen to H2O2. Okay. The second one, glutathione peroxidase. It does the next step. It converts H2O2 to OH negative. And catalase directly convert H2O2 to water. You can see catalase. Let me explain again. You have to learn the reaction. Hana, what is the reaction? Oxygen get converted to water. But learn the three intermediates. What are the three intermediates? 1, 2, and 3. Tell me the name of the three intermediates. The first intermediate is O2 negative. That is superoxide ion. The second is hydrogen peroxide, and third is hydroxyl ion. You know. Okay. Now I'm telling you three scavengers. The first scavenger, SOD, does this conversion. It converts this to this. It converts first to second. I mean superoxide to hydrogen peroxide. The second, glutathione peroxide, does this conversion. It converts hydrogen peroxide to hydroxyl ion. It's very easy if you understand. And catalase, the third one, it directly converts hydrogen peroxide to water. Without this, it directly converts hydrogen peroxide to water, I mean. That is catalase. So this is the three enzymes in front of you. If you know the reaction, it's very easy. It's very easy. So what is the function of SOD? What is the function of SOD? Say quickly. What's the function of SOD? Superoxide dismutase. It converts superoxide to hydrogen peroxide. That's why superoxide dismutase. It is acting. The reactant is the superoxide. The second is the glutathione peroxidase. It is converting hydrogen peroxide to hydroxyl ion. And third is catalase. It converts H2O2 directly to water. So same thing is shown to you here. The three enzymes. You can see this one, and this one. So that is the mechanism of action. So the first is superoxide dismutase. It is present in mitochondria and cytoplasm. It converts superoxide to hydrogen peroxide. You can see this reaction. This reaction, superoxide to hydrogen peroxide. The second one, glutathione peroxidase, it is also present in mitochondria and cytoplasm. Like this only. And it also converts, it converts the second thing, hydrogen peroxide to hydroxyl ion. That is glutathione peroxidase. And third one, catalase, is not present in mitochondria. It is not present in cytoplasm. It is present in peroxisome. And it directly converts hydrogen peroxide to water. It directly converts hydrogen peroxide to water. So that is the three enzymes that taught you. That is how. So if you get a short note on the free radical, can you tell me their definition? The definition, these are the molecules which have single unpaired electron in their outermost orbit. That's why they are very reactive. And they damages the cell. That is the definition. How does they get generated? You write down the three intermediates. So they generated during oxidative phosphorylation, where oxygen get converted to water, and these are the 1, 2, 3 intermediates there. You can write down the reaction. Tell me the types. I told you there are three types. You can write down their names and their formula. That is superoxide, hydrogen peroxide, and hydroxyl ion. What does it causes? It causes damage to carbohydrate, protein, lipid, nucleic acid, damage to everything. And how does it get removed? For the removal, there are three enzymes. Write down the name of the three enzymes. SOD, superoxide dismutase, glutathione peroxidase, and catalase. And which reaction they are conducting. So, which reaction exactly they are conducting, you have to write down. If you're writing all these, you can easily write down 2 pages over there. So, it will be coming for short question. Okay. So, that is the free radical injury. So the summary, I taught you the 4 mechanisms of the cell injury. So I taught you ATP depletion. In ATP depletion, due to ischemia, the cell is not getting the blood, the cell is not getting the oxygen, the ATP is not formed. So 3 things will happen because ATP is not formed. Number 1, sodium potassium pump will not function. So sodium will come in, water will come in, the cell will swell, endoplasmic reticulum will swell, blebs will form. The second thing, anaerobic glycolysis will take place. Anaerobic glycolysis because of which the pH will fall. The pH will be acidic, that will cause clumping of nucleus, the clumping of the chromatin. And third thing, the ribosome will get detached from the endoplasmic reticulum, so no protein synthesis. So these are the 3 mechanisms broadly I taught you. Mitochondrial damage, here also 3 things will take place. Number 1, normal oxidative phosphorylation will not take place and abnormal oxidative phosphorylation take place. So if normal oxidative phosphorylation do not take place, so no ATP formation. That is ATP depletion. And abnormal will take place. That is reactive oxygen species of free radical will form. And third thing, cytochrome C will leak out. That will lead to apoptosis. So these are the three flowcharts you have to draw here. These are the three flowcharts you have to draw here. Loss of calcium homeostasis lead to activation of 4 enzyme. You know the name of the 4 enzyme: phospholipase, protease, endonuclease, and ATPase. That will degrade the phospholipid, protein, nucleus, and ATP. So that is how it is causing the injury. And free radical, I told you a short note. How does they get generated? What is the definition? What does it causes? What are the scavengers? So this is what you have to write down in the mechanism of the cell injury. If it is coming for a long question, you have to write down all 4. If it is the short question, whatever is asked, you have to write down that only.